All right, so we did our urinary system anatomy last week, and we talked about the final physiological action of the system, which is urination or micturition, as hopefully you learned. I still don't know why we have that extra term for it, but that's the extra fancy term for it. So today we're going to look at how the nephron actually makes urine by filtering the blood, as well as how it's able to make urine with different amounts of water in it. All right, so we're going to start with the beginning of the nephron and work our way through and cover some terms and concepts as we go. So there are three processes to make urine. And the same processes that actually make the urine also serve to adjust the composition and volume of the blood if needed, which we'll tackle a little bit at the end. So the first thing that happens is filtration of the blood in the renal corpuscle. So we call it glomerular filtration because the glomerulus is the capillary bed through which it's occurring. And then in the renal tubule, we have tubular reabsorption, which is where we keep things that we don't want to lose. So it's reabsorbing it because it started in the blood, filtered out, and now it's returning to the blood. We talked a little bit about this in terms of calcium reabsorption for calcium homeostasis. So this is where everything that's been filtered out gets reclaimed that we don't want to lose. And then the last step to actually making it urine is tubular secretion, where additional waste products can be added to what's filtered out to get rid of them. So we're going to start with filtration, and then we'll get to what happens in the tubules. So the urine formation is really the primary function of the kidneys, but the kidneys do have additional functions. One of the things that's tied very closely to forming urine is the blood pressure regulation, which we've touched on a little bit already because we talked about the hormones that can affect blood volume and therefore blood pressure. And we've also talked about the intrinsic regulatory mechanisms that the kidneys have in terms of blood volume as well. And that, of course, also affects the composition of the blood as far as like sodium levels and stuff. And we'll touch on that again today to make sure we're clear on how this works from the point of view of the kidneys. A couple of things that the kidneys do that are unrelated to their urine formation functions, they are the source of erythropoietin, which is the hormone that actually stimulates red blood cell production in the bone marrow. And this is because they are pretty much always receiving a good quantity of arterial blood, and if the oxygen levels in that blood drop, they will release more of this hormone. Hold on. Oh my God, sorry, squeaky toys. The other thing that it does, which we have learned about already, is that it's the final activation step for vitamin D, so that our calcitriol is able to have its calcium homeostasis actions, where it enhances the effects of parathyroid hormone, and then it also causes calcium absorption in the intestines to increase. So, filtration, the first step in urine formation. Basically, this is very similar to what happens at any capillary bed in the body. So anytime we have exchange between tissues and blood. It's a little different though, because all of this occurs at a much higher pressure, because when we have exchanges with tissue beds, we're just kind of, you know, diffusing and using other passive processes to move things out of our capillaries and into the extracellular fluid. But now, instead of extracellular fluid, we're moving into this little capsule, right? This space in the renal corpuscle between the parietal and visceral layers, right? It's kind of like trying to fill a balloon. So in order to do that, we need the blood pressure within the glomerular capillaries to be very high, and that pressure encourages the fluid to move out. And it has to be able to overcome basically the size of the space. So we call it the capsular space hydrostatic pressure. And you can really think about this like trying to fill a balloon. The other thing that tries to stop filtration is the proteins and cells that stay in the blood. So the filtration membrane, which we learned about last time, is small enough that the proteins in the blood and the cells in the blood are not able to exit. So they stay in the blood in the glomerular capillary, and they have a pull, an osmotic pull on the water, the fluid component of the blood that's filtering out. And so in order to actually filter fluid out of the blood, the pressure of that blood in the capillaries has to be high enough to overcome these opposing pressures. In order to do that, or I guess the consequence of that, is that we have something called a net filtration pressure. So basically the difference between the pressure that favors the filtration, the blood pressure, and the ones that oppose it has to be positive. And that positive pressure is called net filtration pressure. Net just is kind of a way to refer to what happens when you subtract numbers from other numbers, okay? And so that's what actually pushes fluid out of the blood. And how fast that occurs depends on the systemic blood pressure. So we know that as long as the mean arterial pressure in our arteries is higher than 60 millimeters of mercury, our kidneys are able to keep filtering blood at a normal rate. And so we're gonna talk about everything today, assuming that we have a normal mean arterial pressure, because what happens after that is kind of beyond what we're hoping to learn. So that's kind of, there we go. Okay, so this is just kind of demonstrating that. You don't need to know any of these numbers, but it's nice to kind of see how it all actually plays out. So the blood hydrostatic pressure here is the blood pressure within this capillary bed. And then here's our colloid pressure opposing that. That's again the pull of the proteins and cells, pulling the water back in. And then the capsular hydrostatic pressure is basically just the bounce back from this outer, don't do that, from this outer part of the capsule. So we're trying to fill this space. It's not very big, and so there's going to be pressure as we fill it, right? Like trying to blow air or water into like a very small balloon. And so these are the opposing pressures. So as long as this is higher than both of these combined, we're going to actually filter fluid out. So the way that we calculate the rate at which we're filtering fluid is with something called the glomerular filtration rate. So it really just means how much fluid is being formed within that capsule in a minute, as a sum total of all of the nephrons in both of the kidneys. So GFR is how we shorten it. So it's just, remember this filtration just means moving the fluid out of the glomeruli. So it's just a very self-explanatory. And so basically this is going to be volume per minute in mils or liters. And this rate can be adjusted in order to make sure that the kidneys are always making an appropriate amount of urine, but that we're not sacrificing the blood pressure of the entire body in order to do it. So because we do lose some amount of volume as we make urine, we don't want to decrease blood volume, decrease blood pressure, and then affect the perfusion of the rest of the body. Okay, so this can get adjusted as needed to balance these two opposing needs of the need to make urine by filtering. So filtrate is what we call the fluid once it's filtered out, versus the need to maintain a systemic blood pressure of over 60 millimeters of mercury. So if we have too much blood volume or we need to make more urine for some reason, the kidneys can increase the rate at which they're filtering. So we increase the GFR. This will result in more urine being produced and will decrease the blood volume and pressure. And we know that this happens, like if you're overhydrated or something and you have too much blood volume, you can do this. We know that the opposite is true to a certain extent, although this one is more important. We can decrease the glomerular filtration rate to some extent if we need to preserve blood volume and pressure. So we have a couple of different levels of regulation for this that represent that balance between the need to keep making urine because we have to get rid of these waste products and the need to preserve blood pressure. So we have some local controls at the level of the nephron itself that try to make sure that we're always filtering the enough urine, enough blood. So we call it renal autoregulation because this is how the kidneys regulate themselves. So the myogenic mechanism, myo refers to muscle. This just refers to the tendency of smooth muscle in the walls of a hollow organ to constrict when they're stretched. And this is specifically for the afferent arteriole, which is the blood vessel that supplies the glomerulus. When we have increased blood flow because the blood pressure in our bodies goes up, we will see this small artery constrict. And so what that does is it maintains a steady amount of blood going into the capillaries so that the rate of filtration stays the same. In spite of the fact that the blood pressure coming into the system has changed. Because if it didn't do this, then we would increase the GFR because we increase the blood pressure. But instead, we keep it the same. Okay, so this is about maintaining a constant GFR. And the opposite happens too. So if we have a drop in blood pressure, and that reduces the stretch on the afferent arteriole, we will actually get a reflexive relaxation, which will encourage more blood flow through so that we'll still be filtering the same amount. It won't drop just because the blood pressure dropped. So that's the first one. The second autoregulatory mechanism is by way of that juxtaglomerular complex or apparatus that I told you about last time. So we call this tubuloglomerular feedback because it's based on the contents of the renal tubules affecting the GFR. So basically the juxtaglomerular complex is monitoring the sodium levels, and that's in the distal convoluted tubule because that's the part of the tubule that's connected. If the sodium gets higher, that usually means that the GFR is too high. So we would potentially be losing too much sodium and therefore too much water in our urine. And so this is going to trigger vasoconstriction of this afferent arteriole, and that'll decrease the GFR so that we're not losing too much salt and water. And then the opposite happens too. If the tubule has not enough sodium in its contents, then that is probably because there's a low GFR, and that would mean we're not making enough urine. And so that'll cause vasodilation, increase the blood flow, and that'll increase the GFR so that we're back to making the right amount. Okay, so these are two ways that the nephron is regulating itself to try to maintain a constant GFR. That's usually the goal. Okay, so here's that juxtaglomerular complex again, right here between the distal convoluted tubule and the afferent arteriole. Now there are extrinsic controls as well. So the intrinsic controls are about maintaining a constant GFR. The extrinsic controls are concerned with maintaining a constant systemic blood pressure. And so in this case, we will change the glomerular filtration rate in order to maintain a constant blood pressure. So the two mechanisms that do this, the sympathetic nervous system, so we call this sympathetic control, will change vasoconstriction. So basically when we have a strong sympathetic tone and we kick in our fight and flight reflexes, we will redirect blood flow from the kidneys to the heart, the brain, and the skeletal muscle, because the kidneys are one of our sacrificial organs, where, you know, it's more important to survive right now than it is to make urine. So when we have a high sympathetic tone, we're going to have a reduction in GFR because we're literally getting less blood to our kidneys. The other mechanism that's at play here is the renin-angiotensin-aldosterone system. So when, let me go back a sec. So when we have this tubuloglomerular feedback and we're changing the diameter of our afferent arteriole, we're also potentially releasing renin. So if the blood pressure is low and we have a low amount of sodium detected, then we're going to get renin released from the juxtaglomerular complex, and that renin is going to, oh, I didn't put it in there. We're going to go over this again in more detail later on in this lecture, but just as a reminder, that's going to activate angiotensin, which causes vasoconstriction, which increases blood pressure, and then it also activates aldosterone and antidiuretic hormone. And we'll talk about that stuff more later. So that's how we make the filtrate that gets converted into urine. We are filtering the blood at the renal corpuscle, and then that filtrate is going into the lumen of the tubule, where the reabsorption and secretion processes will actually turn it into urine. So we're going to talk about how we do the reabsorption and secretion. And just like we just talked about the regulations for that world, for filtration, we'll talk about the regulatory effects for those processes as well. All right, so for reabsorption, we're going to be talking very briefly about all of the processes we've talked about for moving things across membranes, because this is all at play again. So just like this was relevant in the digestive system, this becomes relevant again here. So we call them selective transepithelial processes. So most of this stuff is occurring through the epithelial cells that compose the walls of these tubules. And then everything then moves into the capillaries surrounding them, right? Because that's why it's reabsorption, because we're going back into the blood. So when it's through the tubular cells, we call them transcellular. If we're actually, see, there's always a typo. If we actually manage to move things in between the cells, we call it a paracellular process. Now, unlike in the digestive system, where we have a lot of tight junctions to prevent paracellular movement out of the lumen, here we typically have more of, let's just say, less tight junctions. So we call them leaky tight junctions. We tend to have movement of water and electrolytes or ions in between the epithelial cells. So we get more flow by passive processes in the renal tubules than we would in the digestive system. Now, all of the passive and active processes that we've talked about before are going to be at play here. And you're going to see that just like in the digestive system, it's usually a combination of the two. So we'll start by looking at what happens in the proximal convoluted tubule, which is the first part of the tubule, in part because it makes sense to go in order, and in part because most of the reabsorption occurs here. So the proximal convoluted tubule's goal is to reclaim all nutrients that are circulating in the filtrate. So glucose, amino acids, fatty acids, anything that we can use to make ATP should be reabsorbed here. It's also going to reclaim 65%. So you don't need to know the number, but just recognize this is the majority of the salt and the water, the sodium in the water that was filtered out. As well as a fairly large percentage of pretty much all of the other electrolytes and other compounds that were filtered out in the glomerulus. So this is our major reabsorptive site. And just like in the digestive system, the reabsorption of sodium is the most important thing. So 80%, again, you don't need to know the number, but just to demonstrate how important this is, 80% of the energy being used for active transport, so 80% of the ATP being used by the proximal convoluted tubule is being used to pump sodium. So this is using primary active transport, and then most of the other things that are reabsorbed are moving either by secondary active transport using sodium as a co-transport, or because the movement of sodium and these other substances creates gradients for passive transport of water or other electrolytes. So we have primary active transport at the base of these cells. We're pumping sodium out, maintaining a low sodium concentration within the cells. We're doing it by exchanging it for potassium. So we do have a lot of potassium in these cells, which is totally normal. And so the sodium ends up getting pumped into the capillaries as water follows the sodium out of the cells. And then in order to get a bunch of other things in, we have channels in the apical membrane, so facing the lumen, which has the filtrate in it, that are being driven by this gradient that the pumps are creating. So this should sound familiar because this is pretty much exactly how it works in the digestive system as well. So here's our primary active transport. This is where we're actually using ATP. We're moving sodium out of the cell into the extracellular space and then into the blood. Because the sodium is moving out here, we have a low concentration in the cell, and so any sodium in here is going to move down its gradient into the cell, where it then gets pumped out and into the blood. So we can do all sorts of things then to get a bunch of other things reabsorbed at the same time. So chloride tends to follow sodium because of charge balancing. Calcium moves by co-transport. All of our nutrients, amino acids, and glucose, they're going to be following as well with co-transporting. You can see we're getting our phosphorus in here too. Water tends to move by following it down its osmotic gradient. And as we're doing this, we're exchanging hydrogen ions. They're moving into this space so that we're not completely ruining our charge balance, right? Because most of this stuff is positively charged, so we're moving something positively charged back. We're exchanging charges in order to do this. Okay, so all of this is secondary active transport. A little bit of it is some diffusion. The fatty acids, by the way, are just simple diffusion in because they're hydrophobic, and so they can cross the membrane without needing assistance. Okay, so this is explaining that, all right? This explains that previous slide. So secondary active transport of all sorts of organic materials as well as ions because of the sodium pumps, and then other things that are passively following as we're decreasing, or I guess I should say, so water then follows the secondary active transport. As we reduce the amount of water in the lumen of the tube, other solutes, their concentration, relatively speaking, increases. So then they end up moving into the cell because the water left too. So now their gradient favors movement into the cell. And then as I said, oh, I guess I said, yeah, I did say that a little bit. The chloride actually follows sodium because of electrical attraction. So because it's the major negative ion, it follows the sodium, because as the sodium leaves, the filtrate is more positively charged, so then its negative charge favors it moving into the cell as well. This water reabsorption that takes place here is something called obligatory. If something's obligatory, that means it has to happen. And so all the water movement in the proximal convoluted tubule is, it happens every single time we make urine. So this is going to be as opposed to water reabsorption farther down the tube, which is variable. And we'll talk about that when we get there. So what this is showing you is water channels as well as paracellular water movement. It's all by osmosis, because whenever we have water moving, that's what it is. Osmosis is movement of the solvent down its concentration gradient, okay? So basically, as we move more ions and other things into the cell and then into the blood, the water concentration in the lumen becomes higher, right? We're basically diluting the contents of the lumen, this filtrate, and now there's so much water here that it's going to move down its concentration gradient through the cell into the extracellular space and then into the blood. So most of it is following sodium because mostly what we're moving is sodium. Sorry. All right, final thing about the proximal convoluted tubule. So I pointed out all of those hydrogen ions that were being exchanged and moving into what is becoming urine. This is done even more deliberately than that particular diagram showed you. So not only do we use hydrogen ions in order to balance charge for other things, we also want to not lose the bicarbonate in the blood to the urine. So we've talked about bicarbonate before, right? Bicarbonate is the primary way that carbon dioxide is carried in the blood. And even though it's being carried in the blood to get breathed out in the lungs, because of its function as a buffer to maintain pH, we don't want to lose it in the urine. And so we actually reverse the process that, that converts carbon dioxide to bicarbonate in red blood cells to preserve it and reabsorb it. So carbonic anhydrase is the enzyme, always going to be the enzyme that converts bicarbonate into carbon dioxide and vice versa, okay? It takes the equation both ways. Once we convert any bicarbonate in the filtrate into carbon dioxide, it's able to freely diffuse into these epithelial cells. Once it gets back into the, once it gets into the epithelial cells, it'll actually get trapped there because the epithelial cells have carbonic anhydrase inside of them. So they convert the carbon dioxide back to bicarbonate, and then they pump, or they use secondary active transport to move the bicarbonate out of the cell with sodium, and then it ends up back in the blood. That seems like a lot to go through, but I think it underscores the importance of bicarbonate to the body. It's not just a waste product, it has important functions as well. So here's a diagram showing that. So carbonic anhydrase is going to convert the bicarb into carbon dioxide. Carbon dioxide is a small nonpolar molecule, so it freely diffuses down its concentration gradient, which is being maintained because we immediately convert it back into bicarbonate. And here is the hydrogen ion that we've been pumping across in the other diagram. So we're using all those hydrogen ions not only to exchange charge for those sodiums, but also to help us recover bicarbonate. And then the bicarbonate gets pumped out here and it ends up back in the blood. So this is part of how we're maintaining blood pH by recovering this stuff from the filtrate. Pretty sure that was the last thing. Okay, this is the last thing, but they're related. Okay, so in case I had forgotten to say it, but I did point it out. So we're exchanging the bicarbonate for the hydrogen ions. So that means that we are actually secreting acid into the filtrate in the proximal convoluted tubule. Now, there are other acids that can be waste products in the blood. So organic acids are waste products of some of the metabolic processes of cells. And so any of those, if we want to get rid of them, we have to secrete them into urine and pee them out. That's the only way to get rid of organic acids. So not only will the hydrogen ions themselves end up in the urine, but also any other types of acids will end up there too. So lots of hydrogen ions ending up in the lumen of the tube, and as we simultaneously reclaim all sorts of other things. All right, so that takes us to the next part, which is the nephron loop. So the nephron loop has two components. It has a descending limb, which is the first part that goes from the proximal convoluted tubule down into the medulla of the kidney. And then the ascending limb goes back up towards the cortex and then turns into the distal convoluted tubule. They are different from each other. So they're not as permeable to everything as the proximal convoluted tubule is. The descending limb is very permeable to water and some solutes, but honestly don't even worry about that part. And then the ascending limb is not. So water cannot move into or out of the second half of the loop, but there are sodium pumps that actively pump sodium out, okay? And this has very important consequences to the concentration of solutes in the extracellular, which is also called interstitial fluid, of the medulla. All right, so let's see how this works. All right, so an additional 25% of sodium chloride and potassium are reabsorbed here. Again, you don't need to know the numbers. But this is kind of our second biggest reabsorption site, is in this loop, okay? So again, we're using our sodium pumps as our primary mechanism, and we're pumping sodium out of the lumen, and we're getting chloride and potassium, as well as calcium and magnesium by all these various mechanisms. And then the blood vessels are right here, so the reabsorption gets it all back into the blood. Here is a diagram showing you how water can exit the descending limb but cannot move through the ascending limb. So what happens is that the medulla has lots and lots of solutes in the extracellular fluid down here. So the water is going to move by osmosis out of the tube. And as it does that, we have more and more solute inside the tube, right? So because we've moved water out, the remaining stuff dissolved in the water is of a higher concentration. So then we get up here and we start pumping out all of those solutes using lots and lots of ATP. And so now we're diluting what's inside. But because we lost a bunch of water first, what's inside the tube is now a much lower volume than what started, okay? Because we've now reclaimed much more of the water and electrolyte that we didn't want to lose. All right, so this is where it gets a little bit confusing. As we move the water out of the descending limb, we see these numbers representing increasing solute concentration. But because we're pumping the solutes out on the ascending limb, the extracellular fluid also has a very high solute concentration. So we call this osmolarity. And what we see is the osmolarity, or the really high solute concentration. It increases as we go deeper into the medulla. And then it decreases again within the tube as we ascend back towards the cortex, because we're pumping out the solutes that were making it high osmolarity and keeping the water trapped. So because more stuff exits, we have reduced the total amount of filtrate. And we're going to talk about the consequences of this really high osmolarity in a little bit, because its function isn't directly related to the nephron loop itself. It's related to other things in the medulla of the kidney, okay? So the distal convoluted tubule reabsorbs another 10 to 15%. So you can see the numbers are decreasing, right? We get the majority of stuff in the proximal convoluted tubule, and then we get as much as possible in the loop, and then we're trying to get the last of it here. So another 10 to 15% of fluid gets reabsorbed through the distal convoluted tubule, and the distal convoluted tubule is responsive to the hormones aldosterone and parathyroid hormone. So what that means is that the amount of aldosterone present dictates how much sodium is reabsorbed, and the amount of parathyroid hormone present dictates how much calcium is reabsorbed. So these are adjustable reabsorptions here. When we absorb more when we absorb more sodium, we end up leaving more potassium behind. And if we don't absorb as much sodium, we'll end up picking up more potassium. So there's a trade-off here between sodium recovery and potassium recovery. When we get to the collecting duct, we're actually done with the nephron. So remember, the nephron ends in the distal convoluted tubule. Got our little renal corpuscle, proximal, loop, distal, and then it's gonna join up to a collecting duct, which also receives stuff from other nephrons. But the collecting ducts still play an important role in the reabsorption and secretion processes. So what's in the tube is basically urine, but it can still be modified even though it's done with the nephron. So the epithelial cells in the collecting duct, there's two types. Principal cells are all about ions and water. They have channels for sodium and potassium, and they are also responsive to hormones, especially antidiuretic hormone, which dictates how many water channels they have. The intercalated cells are all about acid-base balance. So these guys are all about pH. So they can secrete and reabsorb bicarbonate and hydrogen ions, and control the pH of the urine. Although really what they're doing is controlling the pH of the blood. But that's the exchange. All right, so here's the principal cells. Principal cells are mostly reabsorbing sodium and water and leaving potassium or secreting potassium in exchange for the sodium. And then this is showing you the intercalating cells are secreting hydrogen ions, and they actually exchange it for potassium. So the potassium levels that get of the amount of potassium that's reabsorbed versus secreted is actually dependent on the actions of both types of these cells. All right, so I told you before that the water that gets reabsorbed in the proximal convoluted tubule is obligatory, as in it has to happen. This is true in the nephron loop as well. But the water reabsorption in the distal convoluted tubule and the collecting ducts depends on our hydration status and therefore our hormone status, specifically for antidiuretic hormone, but to a lesser extent for aldosterone controlling sodium reabsorption as well. So antidiuretic hormone impacts how many water channels, again, or aquaporins, are present in the cells of the collecting duct, specifically the principal cells, because they're the ones that respond to it. So the more antidiuretic hormone, the more water channels, and the more water gets reabsorbed, okay? I'm pretty sure I have this somewhere else too, but it does not look like it's on the next slide, so I better say it. All right, so more antidiuretic hormone means more water reabsorption, and that results in more concentrated urine, right? There's less water in the urine, so the urine is more concentrated. And then if we don't have antidiuretic hormone, we don't reabsorb as much water, and we have dilute urine, urine with more water in it. So that would occur like if we're well hydrated. Okay, so this is just kind of summarizing everything we just talked about and what happens at different locations. Again, the take home here is that most stuff gets reabsorbed and secreted, for that matter, in the proximal convoluted tubule. The nephron loop is also doing some reabsorbing, and it's mostly by water and sodium, although of course potassium and chloride go along too. And it's setting up medullary gradient that I'll talk about more in a second. And then the distal convoluted tubule is mostly doing electrolytes and water and secretes acid as needed. And then the collecting duct is responsive to antidiuretic hormone, and so it's the final arbiter of the total volume of urine based on how much water gets reabsorbed. Okay, so one last thing before I do that, because somehow it's completely ended up not in there. You'll notice here that urea pops up a couple of times. So remember, one of the major things that we're doing with urine is getting rid of all of our waste products. And as we talked about a little bit at the end of digestive system, urea is the major waste product of amino acid catabolism, right? This is our nitrogen waste product. So just wanted to actually point that out. Somehow it gets left off, even though it's super important. All right, so now let's look at how this stuff gets controlled. So now we're going to delve a little bit more into what's determining whether or not we're making concentrated urine or dilute urine. And we've talked about these guys before, so now we're just looking at it again a little bit more in terms of what's happening at the kidneys. So remember our renin, angiotensin, aldosterone system, right? So renin is actually coming from the juxtaglomerular complex or apparatus. It's an enzyme, and it's going to get released. There's a couple of different things that can trigger, but that low sodium in the tubule, we can also get it released by the sympathetic nervous system. Okay, so it activates angiotensin, which is already circulating in an inactive form in the blood. Angiotensin needs a second conversion before it's fully active. So it has to circulate through the lungs and get fully activated by angiotensin converting enzyme. This is also called ACE. We can block the action of this enzyme with drugs called ACE inhibitors, so you know, which we do use to control blood pressure. The final active form of angiotensin is then angiotensin 2. It increases blood pressure by vasoconstriction, so decreasing the diameter of blood vessels. It also will decrease blood flow through the afferent arterioles of the nephrons. We typically don't see a major decrease in GFR because the efferent arterioles constrict too. So we're able to maintain a good blood pressure within the glomerulus for GFR, okay? The other thing that we do here is, of course, angiotensin releases aldosterone from the adrenal cortex and antidiuretic hormone from the posterior pituitary. Collectively, they're going to promote water and sodium reabsorption, right? Aldosterone does sodium reabsorption, and we secrete more potassium in exchange, and antidiuretic hormone does water. And this is occurring in the distal convoluted tubule, and this is occurring in the collecting duct. So we get a rapid increase in blood pressure, and then we sustain it by trying to maintain blood volume as much as possible, which helps maintain blood pressure. So here's another example of a chart showing you how all that works. So here's our kidneys releasing renin. The liver, by the way, makes the inactive form of the angiotensin. And then here's our ACE, finally activating it and all of its actions. So increased blood pressure, enhancing sympathetic effects, ADH secretion, aldosterone secretion, and then we get a bunch of reabsorption, and that leads us to better blood pressure. Okay, so that's one of our major hormonal drivers, and obviously it's important in that this has come up several times, right? This is one of our major hormone systems because maintaining blood pressure is so important. Antidiuretic hormone can be released independently of that process as well. So anytime your brain, specifically your hypothalamus, detects dehydration with an increase in osmolarity, so an increase in that solute concentration, it'll release antidiuretic hormone. And again, that's acting on the principal cells in the collecting ducts, increasing the water channels present. Now, what's driving the water reabsorption is that gradient in the medulla. So let me see, no, okay, whatever. So this is just a little diagram showing you the principal cell has a little receptor for the hormone, and when that happens, it puts a bunch of these water channels in the apical surface, and then we're going to have water moving in. So we're going to have a really high, let me go back. All right, so the collecting duct is going into the medulla where the loop is. So remember I said that there's a lot of solutes down here. And so the water is going to move out quite rapidly out of these water channels if they're present, because it's easy for it to move down this gradient. So the nephron loop basically sets up this situation where we have more concentrated solutes in the medulla so that if we need to make concentrated urine, it's really easy to reabsorb as much water as possible. And then once that urine leaves those collecting ducts, at the bottom of the medulla, there's those little papillae, and then it ends up in the pelvis, and now it's urine, and off it goes down the ureters into the bladder. The only hormone that actually acts in opposition to all of that is the natriuretic hormones or peptides. So atrial natriuretic peptide is the most common one. It's called that because it is produced by the atria of the heart, and it stimulates sodium excretion. So we actually lose more sodium, and therefore, because water follows more water, and it does it because it basically just blocks everything we just talked about. So by blocking the renin-angiotensin-aldosterone system, by blocking the release of aldosterone directly from the adrenal glands and blocking the release of antidiuretic hormone from the pituitary, we basically set up conditions where we reabsorb less water and reabsorb less sodium and then reabsorb less water. And this is triggered if we have high blood pressure and blood volume. So basically, we stretch the heart too much because of the high blood pressure in the atria, and that causes the release of this hormone, and that lets us increase the volume of the urine. And so that should decrease our blood volume, which should then decrease the blood pressure. That's the goal. Again, we've talked about all this, but just so that it's written down somewhere separately. Parathyroid hormone, of course, is going to act at the level of the distal convoluted tubule. Whenever calcium levels get low in the blood, it gets released, and we therefore increase reabsorption of calcium from the kidneys. It also blocks reabsorption of phosphate, so that there is more phosphate in the urine when we do this. It increases the release of calcium from bone, right? Resorption. And then it activates vitamin D, which increases the absorption of calcium from the intestines, as well as enhancing both of these things. So again, we've talked about that, so I'm not gonna like dwell on it. Now we're gonna look at the other stuff, the non-urine stuff, very briefly, as we already mentioned it. So again, activates and by final synthesis of vitamin D for calcium homeostasis. And then just as a reminder of all of the important things that calcium does in the body, so why this is important, there's all of that. And then in case I had forgotten to explain a little bit more about erythropoietin earlier, it does make erythropoietin, which stimulates red blood cell production, and it bases it on oxygen levels. The consequence of this one, too, is that if your kidneys stop working, you can actually end up anemic because you're not making enough of the hormone that stimulates your red blood cells to be produced. So that's kind of an unexpected consequence if you don't know that particular role. These guys are all related to all the urine formation stuff we just talked about. So these are secondary effects, right? So this is all during the urine formation process, but it's related to the ability of the kidneys to contribute to homeostasis. So we've already talked about blood pressure regulation, and then the regulation of osmolarity, which again is the concentration of the solutes in whatever, and right now we're talking about in the blood. So this is mostly about how much antidiuretic hormone we're releasing. This also helps us preserve electrolyte levels. And then as we've talked about a little bit already, now the kidneys do then have a role in pH regulation too. not only because we are reabsorbing bicarbonate, but also because we're secreting acids. Okay, so these are all important contributions that the kidneys make to homeostasis. So again, we've talked about all these already as we went through urine formation and hormone regulation, but we can look at them as separately as these key things that the kidneys do. Almost done, but we're going to touch on a couple of things from chapter 26. So chapter 26 is all about fluid balance, electrolyte balance, and acid-base balance. It's essentially tying in the parts from all the different relevant systems about these things. But mostly it's about the kidneys. And so we usually cover it after the kidneys. I'm not going to cover everything in this chapter, and you are not responsible for everything in this chapter. You are only responsible for these sections that I'm covering in lecture, okay? So we're just going to talk a little bit about the fluids of the body, and then we're going to talk about acid-base balance. All right, so fluid compartments. We're going to go over how fluid is compartmentalized in the body. So when we look at the amount of water that makes up a human being, we call that total body water. And the percentage of a human body that is composed of water is relative to their age, their body mass, their sex, and their relevant body fat percentage, okay? The reason why is mostly to do with the fact that adipose tissue has very little water in it, like a ridiculously low amount of water, because it's pretty much just a bunch of triglycerides. Also, in general, our water content does decrease with age. So the older you are, the lower your total body water percentage is. So when we look at the range from infancy to being rather elderly, we see that the total range possible in people is between 75% and 45%. So infants tend to run around 75%, and it slowly decreases over time, and the elderly are going to be more likely to be more like 45%. When you look at the composition of the body, we see that it's not evenly distributed. So different organs and tissues have different proportions of water as well, which you might have been able to guess since I just talked about how adipose has very little water in it. So here's some examples. So for example, the brain and the kidneys are 85% water. Those are some of the more hydrated tissue types that we have. On average, most of our organs are about 70%. And I think that's where we get that whole 2/3 of your body is water thing, just like the ocean, which is just a coincidence. You know, like 2/3 of the surface of the earth is covered by water. Blood is only 50% water by average, which we knew, right? Because we know that like 45% of blood is red blood cells. So even though it's liquid, it doesn't actually have that much water in it. Bones are usually about 25% because there's so much mineral in it, and because teeth have even more mineral, they're not even 10% water. And I always forget to actually look up, yeah, it's not on here, what adipose is, but I assume it's less than that. It's gotta be pretty low. Because basically the only water in there is like what's in the nucleus of each cell and like the capillaries surrounding the adipose. Okay, again, you don't need to know the numbers, just understand that it's different by tissues. Sorry, I get all excited and then I forget to say that part. Okay, this is what really is important. Fluid compartments. So we divide all of the fluid in the body into two main compartments. There's the intracellular fluid, so the fluid inside of the cells, which makes up 2/3 of all the fluid in the body. And then there's the extracellular fluid, which is everything else. And you'll see we do abbreviate them ICF and ECF. Now, there are additional subcompartments of extracellular fluid that we're concerned with. We don't worry about the intracellular. It's just a blanket intracellular fluid. But there's two major extracellular fluid compartments. There's the interstitial fluid, which is what's between cells. And then there's the plasma, which is what's in the blood. The reason why we have these definitive compartments is because there are boundaries between them. There are plasma membranes separating these compartments, right? So the intracellular fluid is everything within the plasma membranes of individual cells. Extracellular fluid is everything outside of those plasma membranes. The interstitial fluid is what actually surrounds the plasma membranes of cells, whereas the plasma volume is what's contained within blood vessels. Now there are other tiny ones. There is an other, and we don't get too worried about it because they make up a very small percentage. So intracellular fluid, that's about our 2/3 right here, 66%, is what's inside these cells. And then the extracellular fluid is everything else. So the interstitial fluid makes up the majority of everything else. Most of our extracellular fluid is interstitial. That's all this stuff here. And then our plasma volume is the next. You can see it's not a ton. It's just the next largest compartment. And then this is other. Other is stuff like the water inside your eyeballs. It's your cerebrospinal fluid. It's the liquid inside of your joints, synovial joints, all that stuff. Small spaces. All of our body fluids are water-based solutions. So water is our solvent. It is the thing present in the largest volume. And the solutes, the things that are dissolved in the water, can be divided up between electrolytes and non-electrolytes. So electrolytes are the things we also call ions, right? This is like our sodium and our potassium and our chloride. The non-electrolytes are things like glucose and amino acids and waste products like urea, and technically things like carbon dioxide and oxygen and stuff like that too. One of the other things that makes intracellular fluid so very different from extracellular fluid is that the composition of solutes is very different. So intracellular fluid is very high in potassium and phosphate and proteins, okay? That's these guys right here. The extracellular fluid is very high in sodium and chloride, and everything else is kind of lower amounts. So you can see like bicarbonate's pretty low in all of them. Obviously it's pretty low potassium in our extracellular fluids, as well as pretty low phosphorus. Calcium's pretty low everywhere, as well as magnesium, although both of them are in slightly higher proportions in the intracellular fluid. And then protein is basically non-existent in the interstitial fluid, highest in intracellular, and then intermediate here in plasma. Again, you don't need to know everything on here, just kind of focus on what I actually wrote down. The key here, though, is that the extracellular fluid is different from the intracellular. And the important parts are the potassium, the sodium, and the chloride. And we've talked about why that is, right? We have talked about our sodium potassium pumps, our sodium potassium gradients, and how we use that to make electrical things happen. So in spite of those gradients, we know that the fluids in general tend to be neutral. So when there's charge differences, it's just at the membrane. It's not the whole fluid inside the cell or outside of the cell. And we know that the concentration gradients and the charge gradients are maintained almost exclusively by sodium potassium pumps. There are other things at play here, but almost exclusively these pumps. So in order for fluid to move between these compartments, it's typically a passive process. We are not really actively moving water or like the whole solution in an active way. Instead, it's all about pressures and gradients. So the hydrostatic pressure is the general term we use for the pressure of a fluid against its boundaries. So when we're talking about the blood, that's blood pressure. But we're talking about other fluids, that's other types of hydrostatic pressure. However, the blood pressure is the one that's super important because it's the one that's typically the highest, and it results in the movement of fluid out of the vascular space. This is how we filter things through all of our capillaries. So not only does this apply to glomerular filtration like we talked about today, but it also applies to all movement of fluid out of capillaries in the whole body. Water moves passively down its osmotic gradients. So osmosis is always about the solutes that are trapped in the fluid compartments. If you have a solute that's trapped, then the water is going to follow its osmotic gradient in or out. And for the most part, this is between the extracellular compartments. So most of the movement of water is between the interstitial and plasma spaces. We only really have movement into or out of the cells and the intracellular space when the cells get a little dehydrated and need some water, which really only occurs if, just ignore that part, pretend I didn't say it. Don't worry about that. Okay, so solutes can move actively, right? We know we have these pumps because we're maintaining concentration gradients. So remember that whenever we move against a concentration gradient, it's an active movement, which literally means that we are using energy. And passive movement, while it doesn't require energy, still requires permeability. So things don't just move passively because there's a gradient, things move passively because there's a gradient and there's a pathway. So again, we have to remember that other than things that can move by simple diffusion, which is basically non-polar small things, it's pretty much just oxygen, carbon dioxide, oh my god, I'm writing carbon dioxide twice, and like some small lipids. Everything else is going to be facilitated, right? So that means that we need protein channels and pumps. Or not pumps, hold on. Carrier proteins, that's what I was looking for. So here's glucose, for example. It can't cross the plasma membrane unless we have carrier proteins that are allowing it to move through, right? It's going to move passively down its gradient. There's no expenditure of energy, but it still requires a pathway to take. The book likes to talk about the edema here, so I just wanted to point it out. So edema is defined as accumulation of excess water in tissues. This is almost always increased amounts of interstitial fluid. So in the fluid around cells. And anything that favors the movement of fluid out of the plasma space or out of the intracellular space will cause edema. So usually this is caused by increased vascular permeability, which means your blood vessels are a little too leaky. This can also be caused by a decrease in the colloid osmotic pressure in the blood. So what this means is when you have less proteins in the blood, there's not as much draw to hold the water in, so it'll leak out into the interstitial space. It can also be caused by an increase in the hydrostatic pressure. So an increase in your blood pressure can potentially cause fluid leakage as well. And we see this a lot in like people with hypertension, people with heart failure, because their blood pressure is really high, trying to continue to deliver it to all the tissues. And the consequence of that is that we can have some of that fluid leaking out and getting trapped in the interstitial space as edema. Last thing we're going to talk about today is acid-base balance. So we have touched upon all of the concepts we're about to talk about, and now we're just going to put it together as an overall acid-base homeostasis of the whole body. All right, just to make sure, I hope that this is just a review of pH again. We touched on this way back when, but I want to be absolutely sure that we are comfortable with these terms. So remember that pH is about the concentration of hydrogen ions in a solution. So it is a measurement of acidity, and an acid is defined as a compound that releases hydrogen ions, and the pH scale runs from 0 to 14. 7 is neutral. Anything less than 7 is acidic. Anything greater than 7 is basic or alkaline. They mean the exact same thing. So an acid releases hydrogen ions and decreases the pH of the solution to make it more acidic. A base is a compound that releases hydroxide. Hydroxide ions combine with hydrogen ions to make water. So hydroxide ions neutralize hydrogen ions. Bases can also simply bond hydrogen ions and take them out of solution. Either way, we have less hydrogen ions, and so we increase the pH to make it more basic. Now, our blood and tissue fluid, so all of our extracellular fluid, has a normal pH range of 7.35 to 7.45. So the average pH in the human body is 7.4, which is very slightly basic, right here. Just a little basic. And it needs to stay in this very narrow range because otherwise, bad things happen. If you want to know more about acid-base stuff, here's a couple of nice sites to start out with. 1 second. Oh, sorry. Okay, where was I? Oh yeah, so bad things happen if you get out of this very narrow range. And we have terms for this. Okay, so if your blood becomes too acidic, which means it becomes less than 7.35, we call this acidosis. You'll also see it called acidemia, but I'm sticking with acidosis. If your blood becomes too basic, so greater than 7.45, it's called alkalosis. And it is bad either way. Too much acid and too much base are both bad. And this is because proteins and enzymes in particular, their shape is influenced by the pH of their environment. And the pH, most of the proteins in our body work within this pH range of 7.35 to 7.45. So when we get out of that, their shape distorts. Call it denatured. And a denatured protein does not function. So if our enzymes are not functioning well, then we're going to have major problems with our metabolic processes. We get acid mostly as waste products of metabolism. So carbon dioxide, of course, is a major waste product of metabolism. Got it, delete that. So remember that we are making that as a consequence of oxidative phosphorylation, and we are carrying it in the blood mostly as bicarbonate. Lactic acid is the waste product of just doing glycolysis without the rest of the steps. Fatty acids and ketone bodies released during fat metabolism still count as acids. And then phosphoric acid can get released during protein catabolism. And so that's going to be acidic as well. So all of these things can potentially lower the pH in our fluids and cause problems. Now, what I want to note here is that we don't really influence the pH of our body with what we eat or drink, okay? We really don't get much acid entering our body from our foods or beverages. And I say this because they sell alkaline water now, and there's absolutely no point in drinking it, okay? Just gonna point that out. You do not change your pH with your diet. All right, moving on. So how do we make sure that our pH doesn't change when we're producing all of these metabolic acids? We have 3 steps, and they are sequential. And what I mean by sequential is this one happens really fast, and then this one can respond next, and this one is slow, and so it takes longer, but it works really well, okay? So they happen in time. So there's these 3 different systems we have in place to regulate the pH. We talk about it as mostly our blood, but technically it's all of our body fluids. So we have chemical buffers, and then these 2 things, which are basically how we can breathe and change our pH and how our kidneys can affect it, these are called the physiological buffering systems. So we mentioned buffers before, right? A buffer is a substance that binds hydrogen ions and removes it from solution as hydrogen ion concentration rises, or releases hydrogens into solution as its concentration falls. And that's why we define a buffer as a chemical that maintains a relatively constant pH in a solution in spite of changes in the hydrogen ion concentration. A buffer system is typically a mixture of both an acid that releases hydrogen into solution and a base that binds it, which makes it very effective at resisting changes in pH whether an acid or a base is added. Buffer systems function to restore and maintain normal body pH. There's buffers outside the body too, but for our purposes, it's all about the fluids in the body. So there's 3 major buffer systems, and they're important in different fluid compartments. We'll go through them one at a time. So the bicarbonate buffer system is considered the only important extracellular fluid buffer. Remember that this is driven by carbon dioxide and the fact that most of the carbon dioxide in the blood is carried as bicarbonate. So we have this carbonic anhydrase, which drives this fully reversible reaction where we can combine carbon dioxide with water. We make a base called, or an acid, oh my God, an acid called carbonic acid. And when carbonic acid releases a hydrogen ion, we get a bicarbonate ion. Bicarbonate ion can bind a hydrogen ion and make carbonic acid. And then the carbonic anhydrase can convert that carbonic acid into carbon dioxide, and we can breathe it out. Or it can actually be excreted in our kidneys. So in both our plasma and our interstitial fluids, the bicarbonate system is the major system that maintains our pH, keeping it at an average of 7.4. This is just a picture I like to show because baking soda is bicarbonate, and when we add it to vinegar, which is a weak acid, we get a bunch of bubbles. Guess what the bubbles are? Carbon dioxide. Okay. Anyway, yeah, so we're gonna touch on that again in a minute. It's super duper important. Inside of our cells, we actually have a lot of phosphorus in the form of phosphoric acids. And these guys are able to operate very, very similarly to how the bicarbonate system operates. So this guy can release an acid when it needs to, and this guy binds the acid and takes it back. So this is a fully reversible reaction where we have an acid and a base buffering the pH. This is the major buffer of the intracellular fluid inside of cells. And this is also important in the lumen of the renal tubules. So we didn't talk about it, and we're not going to worry about it as far as the kidney function, but this is one of the major guys that helps the kidneys do their acid-base thing. Last one is the protein buffer system. So proteins, which exist in both plasma and cells, but not in the interstitial fluid, are really good buffers. And they make up the vast majority of buffering inside the cells. So in the intracellular fluid, the protein buffer systems are the most important. The phosphorus thing does its thing, but the proteins are more important because there's way more proteins than there are phosphorus. In fact, the buffering power of the intracellular fluid in the cells accounts for 3/4 of the chemical buffering in body fluids total. So that makes this way more important for total body buffering compared to the bicarbonate system. The reason it's able to do this is because every single amino acid in every single protein has a side group. And some of those side groups can act like acids and some of them can act like bases. So some of them are able to donate a hydrogen and some of them are able to bond to them. And so when a bunch of the amino acids in every single protein in every single cell in the body is able to do this, we have a very powerful buffering system. In addition to that, we do have plasma proteins that can do the same thing in the blood. They're not going to be as important in the blood as the bicarbonate system, but they are still there, and there's an interaction with the bicarbonate system because carbon dioxide can bind to the proteins and make those carbamino compounds. So for example, we can have carbaminohemoglobin. So there is an interaction between these two systems. Okay, so those are our chemical buffers. Now the chemical buffers are pretty good at maintaining blood, the pH of the different body compartments most of the time, but they're not perfect. And so we have these additional systems in place to kind of supplement that. The first one that's able to kick in is the respiratory control. So we've talked when we were in the respiratory system about the fact that the major driver of breathing rate and depth is carbon dioxide levels in the blood. We have chemoreceptors that are monitoring this. And so as we're altering our breathing rate and depth for the purposes of making sure that we're getting rid of enough carbon dioxide, this also serves to control the pH levels, especially in the blood. So generally speaking, when we add carbon dioxide to body fluids, the ultimate result is increasing the amount of hydrogen ions and decreasing the pH. We're always producing carbon dioxide, so all we have to do is breathe off less of it to increase the amount, right? So this occurs because this equation gets forced towards the bicarb, forced towards the bicarb level if we keep adding a bunch of carbon dioxide. When we increase it here, everything kind of moves down the gradients and we end up with more of this, okay? So because of that, more carbon dioxide means more acid and vice versa, less carbon dioxide means less acid and a lower pH. So when we have increasing carbon dioxide levels and a decreasing pH, so acidosis, our chemoreceptors trigger an increase in ventilation and we breathe off more carbon dioxide. So that's how respiration can control that. The opposite occurs too if our carbon dioxide levels get too low and the pH becomes too alkaline, we will breathe less. So we have a decrease in ventilation, and that lets carbon dioxide build up and add acid to the system to bring us back to normal. Respiratory control is quite fast. We can correct blood pH changes in just a few minutes thanks to the brainstem respiratory centers. But that also has a consequence. So anything that interferes with our ability to breathe normally can actually cause an acid-base imbalance. So respiratory acidosis is when we are not breathing off enough carbon dioxide. So we call this hypercapnia, which just means an increase in the CO2 levels in the blood. So basically anything that makes it harder for you to breathe so that you're not getting rid of enough carbon dioxide can cause respiratory acidosis. Respiratory alkalosis is actually a lot less common, but that's when you're breathing too much and you're getting rid of too much carbon dioxide, which we call hypocapnia. And this is actually what happens when somebody hyperventilates. So if somebody's having like a panic attack and they're breathing really, really fast, they will cause respiratory alkalosis and hypocapnia, and it'll make them kind of lightheaded. And this is why having them breathe into a paper bag is helpful, because now some of your carbon dioxide, you're rebreathing it, trapped in that paper bag, and that helps prevent your carbon dioxide levels from getting too low. Here's your reminder flowchart of how carbon dioxide and blood pH have negative feedback loops controlling respiration. This is the specific one, and you can see that we can see the respiratory control of pH, both for decreasing and increasing the pH, how our respiratory rate can be stimulated to change to basically control the bicarbonate buffer system, right? We are modifying the bicarbonate buffer system with our respiratory control. Okay. Last thing is the kidneys. So the kidneys, through the mechanisms that we talked about already, are capable of neutralizing more acid or base than either of our other systems. And that's because we can either make more bicarbonate or excrete more bicarbonate as needed. So we can actually replenish our bicarbonate stores through our kidneys and renew that chemical buffer. And because those organic acids can get secreted in the renal tubules. It's the only way to get rid of acids that are not carbon dioxide. That's what non-volatile means. Okay. So the respiratory system can only get rid of carbon dioxide-based acid, but the kidneys get rid of all the other organic acids that we're making through metabolic processes. So here's the same thing. We looked at this before, but now we're looking at it from the point of view of secreting hydrogen ions and reclaiming bicarbonate. So the only problem with renal regulation is that it is slow, takes a couple of days to kick in if it needs to make major adjustments, okay? That's really its only downside, is that it's a little bit on the slow side. So I'm just going to introduce you to what we call disorders of acid-base balance. Your book talks about like how we can adjust for them and stuff, and I'm not going to ask you about that. So don't worry about compensation or origin of these things. I just want you to be a little bit familiar with it since we've already made it this far. So acidosis is, again, remember what we call it when there's too much hydrogen ions. The problem is when it starts diffusing into the cells. So because we get a buildup of this in the plasma and the extracellular fluid in general, we end up with a concentration gradient where we start getting more acid entering the cells. Then what happens is that potassium leaves the cell to maintain the charge balance. Potassium's our major intracellular cation, or positively charged ion. And so as hydrogen goes in, potassium comes out. Now the hydrogen gets buffered by those intracellular proteins. So we don't actually have acid in the cells themselves necessarily. So what ends up causing a problem is losing our potassium. Okay, we lose our potassium, the hydrogen ions get buffered, so now we don't have as much positive charge inside the cell. And the result of that is hyperpolarization. It becomes more negative. So in cells that actually rely on their electrical activity, it becomes harder to get electrical impulses. Action potentials become more difficult to stimulate because now these cells are more negative. So what we see is the result of neurons and muscle cells being harder to stimulate. We see depression in the central nervous system, which manifests as confusion, disorientation, and it can potentially lead to coma and death if it's bad enough. I know this is such a bummer to end it on, but that's what happens. Okay, so alkalosis is the opposite. So with alkalosis, we have decreasing hydrogen ion concentration. We get hydrogen then leaving the cells down their concentration gradient. And somehow this results in even more potassium entering the cells. That's bad. And I feel like maybe it's wrong. Gotta check that. But anyway, the end result here is the opposite. So now we have extra positive charge inside the cells. So we're actually depolarizing them. That makes the muscles and neurons more excitable, more likely to charge. And so what we see is the opposite. We see hyperactivity. We see muscle spasms leading up to like tetanus. So you'd be getting like major cramps and stuff. Potentially convulsions as your neurons are firing too much in your brain, and ultimately you could die of respiratory paralysis. So basically, if your diaphragm undergoes tetany, it contracts and doesn't relax again, and now you can't breathe. So that's bad, obviously. All right, so here's a nice little diagram kind of laying them out. Interestingly, the effects are opposite, but some of the symptoms are the same because overstimulating and understimulating all of your electrical systems can have similar outcomes, which is just kind of weird, but that's the way it works, okay? So just so you know, that's what actually happens when your pH is wrong and your diet has nothing to do with it. And so yeah, I would not recommend spending money on expensive alkaline water because it is useless. That's my take home. Anybody have any questions? That's kind of the renal system and related homeostatic mechanisms in a nutshell. I'm gonna get out of that. Stop doing that. If anybody has any questions, please email me. We can always, GFR, okay. Sure, hold on. Give me a second, let me go back to it. Control, filtration pressure, okay. All right, so, okay. So with GFR, when we look at the regulation, what we're looking at is two things in balance. So the goal of the kidneys is to make sure that we always have enough blood flowing through the glomerulus to get enough glomerular filtration. So we're trying to maintain the glomerular filtration rate. And so these two mechanisms, the renal autoregulation, are all about maintaining GFR regardless of what happens to systemic blood pressure, basically, okay? So the myogenic mechanism is all about how changes in blood pressure aren't going to change GFR. So basically, because the smooth muscle of the afferent artery, or arteriole, sorry, has a reflexive action that works in opposition. So if blood pressure goes up, the arteriole constricts and maintains the blood pressure. If blood pressure goes down, then it relaxes and still maintains GFR. The tubulo-glomerular feedback is how the juxtaglomerular complex responds. So basically this is analyzing the filtrate or the urine that's already forming. So this is detecting it in the distal convoluted tubule. And basically if you're making too much urine, we decrease the GFR. Too much urine as demonstrated by having too much sodium in there. And if you're not making enough urine, so the sodium level's too low, then we're going to increase the GFR. And we're going to do that by controlling, again, the blood flow through that afferent arteriole into the glomerulus. Okay, so this is how the kidneys make sure that we're maintaining GFR. Now, we have, in opposition to that, the extrinsic controls. So the extrinsic controls, they're concerned with systemic blood pressure. So they will sacrifice kidney function in order to maintain blood pressure to the entire body. So they will regulate GFR in a way that affects it, like negatively impacts it, but only when blood pressure gets really, really bad. So the major regulators here are the sympathetic nervous system. So when we have increased sympathetic tone, so think about what happens when there's a lot of adrenaline in your system, right? We want to prioritize blood flow to the heart, the brain, and skeletal muscle. And so when we do that, we will sacrifice some blood flow to the kidneys. So we're going to decrease blood flow to the kidneys, and that's going to decrease GFR. The other one is that renin-angiotensin-aldosterone system. So when we have low systemic blood pressure, which also gets detected by that juxtaglomerular complex, we get a release in renin. And the renin, of course, activates, that's the R, right? Activates the angiotensin and the aldosterone. And that's going to result in, see, I really should have put that there. That's going to result in vasoconstriction. Angiotensin does that, right? So we get vasoconstriction, and that will decrease GFR. And then we will stimulate the kidneys also with aldosterone and antidiuretic hormone, but this is the big one that affects this. So in both of these, what we're getting is vasoconstriction, which decreases blood flow to the kidneys, and that results in a decreasing GFR. And so the goal here is to maintain systemic blood pressure, even if we're not necessarily preserving full kidney function at that moment. Okay, does that, is there anything else to clarify that? I don't know if that answered the question. Any other questions before we go? We'll probably have some time next week as well. So if we have additional questions on this stuff, we can address them either at the beginning or the ends of the reproductive lectures. So just let me know, or just send me an email. if you have specific questions over the weekend. And we'll talk next week as well about the last like mid-semester exam and then the final exam too. Okay, we'll tackle all that next week. All right, so it's weird when there's a holiday. See you guys next week.